Welcome to the second tutorial of BacStalk, showing how to do cell stalk detection. So in this case we're using a data set of Hypomonas neptunium. It's a time series which has also been shown in the manuscript, which has phase contrast images and images in the YFP channel. So then we're opening BacStalk. We already imported the files. If you want details on this, please have a look at the first tutorial about importing images and assigning metadata. So in this case, we immediately go through cell stalk detection, which shows us then the second panel of the graphical user interface. Here one sees the active image, and there is a slider which can walk you through the time series. So in this case, we'll go to the last frame, so the 11th time point, which is also shown over here. One can then choose to see the phase contrast or the YFP channel. Can also use different color maps to display this. And the intensity scale is scaled automatically, but one can also tune this. So now the next thing to do is to go and process the images. So there are multiple options here. One can either process one image at a time or process the entire time series, and this is what we will do, but we will only process a part of the image. So we will set a region of interest here, which we can then move. And then it's important to apply this region of interest to all the images in our time series. Now one can process the images, so in this case we will process all the images. So the parallel pool is started, which then speeds up the processing time clearly. And then processing of images consists of two different steps. So first of all there is the drift correction, and then later on there is the processing of the image, which detects the cells and the stalks. If the processing is done, one can see this because of cells appearing in the cell list and of an overlay appearing on the image. So the cell list displays cell IDs, which are also shown then on the image. It already gives the area of the cell, and it shows if the cell has a stalk. And the stalk is always assigned to the mother cell, not to the bud. Or if the cell became deleted because of some reason. So in this case, the cell was deleted. One can overrule this, for instance by clicking here, and it was then also showing us that it was automatically deleted. So we can restore the zoom and have a look now at this cell, cell number 15. So we can select it either here or click over there, and we get a zoom in of this cell. So this cell has formed a bud. The bud is indicated by the dashed line, and the mother cell is indicated by the solid green line. The stalk is indicated by the red line here, and the medial axis are the yellow lines. So one can now manually overrule this. So if one does not trust this cell, one can delete the cell, either by clicking on the checkbox, by clicking on the cell outline, or by clicking on the number, and one can re-enable this. Now there is the option to use the pen to navigate a little bit and to make some measurements. And these measurements can be done or indicated both in pixels, which is really useful for the parameters for segmentation, which will be explained in a more advanced tutorial, or in microns. Then one can delete this again. Then a scale bar can be added, and one can give the size in microns for this. And then a screenshot can be made, and for this screenshot one can choose to display the overlays as made by BacStalk, or to make one without overlays. Then one can uncheck this box here. So a screenshot can be saved there, and one has the option to either save screenshots in PNG or as EPS files, and EPS files are of course very handy for using in Illustrator later on to be able to change things. So I can save this, and then it opens as a new panel. Now we have of course this time series here, in which, let's delete the scale bar first, in which our cell starts off as a swarmer cell, and then grows a stalk after some point, which is indicated in blue if it's not connected to a bud yet, and then grows a bud in the end. and we can see that in the end the YFP signal is in the bud, whereas in the beginning it's still in the mother cell, of course, at the other pole. So in this case one would like to track the cells, whereby BacStalk connects the cells in all the separate images to each other and assigns one ID for this same cell throughout time. So one clicks track cells, and then a track ID also appears next to the cell. One can click on the track ID, and this brings one to the last image in this track. So this was the tutorial on cell and stalk detection. The next tutorial will be about analysis.